Hi, everybody. My name is Laura Maker. I help organize this along with Dr. Smith and Dr. Jessup. We do it every Friday from noon to 1. I just wanted to say we have a completely full schedule for this fall. So if you want to talk next spring, go ahead and email me so I can put you on the schedule for next spring. But we have some great speakers coming up, so be sure to check your email box.
Thank you. So it's my pleasure to get to introduce Dr. Matias Kirst. I've known Matias since he was a postdoc at Cornell when I was a graduate student there. But Matias started his career in Brazil at the University of Santa Maria in 1997. He got his bachelor's, and then he got his master's at the University of Viçosa in Brazil. Viçosa.
Yeah, that'll do it.
Okay. In genomics. And then he went on to the very prestigious program at North Carolina State working on genetics and genomics, which I think is where he got a lot of his statistical influence and also learned a lot about gene expression and really applying genomic tools to breeding. He did a postdoc at Cornell in 2005 with Ed Buckler, 2004. And then after that, he joined the faculty at the University of Florida in the School of Forest Resources and Conservation. So Matias has had a pretty impressive publishing record. He has 40 publications, including those in science with 2 genome papers, PNAS, genetics, crop science, PLOS ONE, etc. But the thing I really appreciate about Matias's work is he's much more quantitative and statistically oriented, I think, than a lot of the people applying genomics and breeding-based tools, and he integrates those things really nicely with gene expression. There's few leaders still in the field of genome-wide prediction and selection, and that was a real big interest in why we invited him to come speak to us. So please, Matias.
All right. Wait, just before you get started, would you ask if Lubbock is on, can they hear? I know Lubbock was going to join us.
Lubbock, can you hear us? Can we hear you?
I asked Judy, she said she didn't think they were on yet. They weren't on yet. So I can text her.
I mean, usually if it's just showing us on the screen, nobody's dialed in.
Well, I just talked to Lubbock this morning. I knew there was 2 who were supposed to be joining us.
We're supposed to be joining us. Okay. So if I interrupt you in a few minutes, it's to
No worries.
connect.
All right. Thanks, Seth, and thanks for the invitation. You know, I always tell my students that are doing breeding that the great thing is that there's lots of jobs out there. There's a ton of jobs out there. They're all getting hired, 6 months before they graduate. The problem is it seems that Texas A&M is graduating more breeders. Right? So if you guys continue with a group like this, you probably will be probably occupying a lot of these positions out there. So this is really the biggest group of breeders that I've seen in any other university. So this is really very impressive. I'm going to talk about genome-wide selection, and maybe we can call this genome-wide prediction. I prefer to use the term genome-wide prediction because we don't necessarily want to use this as a tool to really identify, necessarily do early selection. We might be able to use this, for example, to do things like what I call mate pair location, and I'll go into this a little bit more a little bit later. But one of the points I want to make here is that, as I was telling Tom Byron this morning, we sort of, Seth and I, we come from a generation where we've done QTL analysis, where we've done association genetics, where we've done a lot of sort of interesting things where our primary aim was to integrate the molecular information into the breeding. And I have to admit that I had a really very hard time until quite recently to come to a breeder with a straight face and tell him that our molecular tools were going to help him. Okay, because really QTL analysis was great to identify certain genes of large effect, but really the success of using that information in breeding has been somewhat limited. With genome-wide prediction, I think we have a real possibility to make an impact. Okay, we have a real possibility to truly begin integrating these molecular tools, especially for selection and breeding of complex traits controlled by large number of genes of small effect. So I'm going to talk a little bit about early selection using genome-wide prediction. The first few slides, I'm going to spend a little bit of time talking about genome-wide prediction, how the models work in the sense of sort of giving a background information to people that are not very familiar with it. But then I'm going to show a few examples of how we apply it and how we want to use it now to do what we call the mate pair location. Okay. So, first of all, let me just acknowledge a few people that have been heavily involved in all the work I'll talk about. First of all, a couple of students, Marcio and Patricio Munoz. Patricio is now faculty, Marcio has his own startup company. There are a number of people at Iowa State, Dorian and Rohan Fernando, as well as Gustavo de los Campos, University of Alabama, that have really worked with us very extensively in developing a lot of the things that I'll talk about. So I really have to acknowledge them beforehand, as well as Marcos Rezende in Brazil at the University of Viçosa and Dario Grattapaglia. Funding for a lot of this work has really come from a number of sources, but really primarily USDA. Okay, so I work with tree breeding. Okay, and you're looking at this little branch here, and I got to say that this thing is probably sitting about 30 to 40 feet above the air. Okay, breeding trees is not really a very pleasant activity. You have to usually stand pretty high in the trees. It is kind of dangerous. It's a complex process, and it's very expensive. Okay. Another big problem about tree breeding is that it takes a long time. Okay, and that's why I say that usually tree breeders, they have plenty of time to think about more efficient ways of doing things, because we just, just the process of breeding, making the crosses and waiting for the seeds to develop can take up to 10 years. The step of testing the material that you have in the field may take 6 to 8 or even more time. And then, of course, when you make a selection, then to propagate that selection, you might take another 8 to 10 years or more. Okay, so we're talking about almost a quarter of a century to just run one round of breeding and selection, which means people like Tom and I, we got plenty of free time. Okay, now breeders have come up with ways that are more efficient of doing propagation and breeding. I'll mention those in a minute. But really, testing has remained a very significant bottleneck. Okay. Now, probably most of you working with agricultural crops, you'd say, well, for most of the challenges that we have out there, we probably can resolve them because we can breed so fast for our crops. Okay. But we are starting to sense a certain level of pressure from society in order to actually be able to provide biomass at lower costs and faster. In other words, we are really under pressure to accelerate the pace of breeding of these trees. Okay, and part of the reason for that, of course, is this idea that you probably would not want to use maize to produce biofuels, but rather you would try to use things like woody biomass. And this higher demand for wood is not a projection, is not a fantasy. This is a biomass plant just outside of Gainesville, where I live, that essentially is burning pine trees. Okay, they essentially get the pine trees, they chop them off, they throw them in this massive fire. They move they produce steam and then move the turbines and produce energy. This biomass plant is producing about 40% of the energy consumed in Gainesville now. Okay, and the expectation is that these plants will become more and more frequent across the country. So that's one thing, is demand for biomass. It's increasing, and we need to be able to provide it. The other issue is that little problem called climate change, right? So this is the cold hardiness zone map from the U.S. back in 1990, 2006. And you can see that, southern states here, they were mostly zone 7 and 8. 16 years later, they're mostly zone 8 now. And that in itself, you might say, well, I can run 2 or 3 cycles of selection every year with my agricultural crop. But when you're working with trees, you have a really serious issue on how you're going to be able to develop the materials that will be able to tolerate these very dramatic changes in climate that are occurring. And this is just an example of those consequences. Okay, this is an area in British Columbia. The red trees that you see there those are not the fall collars. Those are pine trees that are dying. And they're dying because of a mountain pine beetle that essentially carries a fungus that kills those trees. Now, until quite recently, the fungus and, let's say, the beetle has lived with these forests without causing many issues. What would happen every winter would be that the population of the larvae would die out, large numbers of those individuals, of those larvae. What is happening now is because the winters are not as severe as they were before, they're actually surviving in much greater numbers and are now starting really to attack these populations with much higher frequency and intensity than they did before. So the situation right now looks more or less like this. All this region that you see in red here in northern Canada, our trees is an area where essentially between 90 and 95% of all the trees are dead. Okay.
Wow.
So when you have that kind of pressure, how do you respond if your breeding cycle takes 25 years, right? So we really need to be able to develop something that will be faster. And then finally, we have a bit of an issue in Florida with invasive species. Estimates is that we have roughly about 100 new species moving into the state of insects every day. Okay, every single day, 100 new species of insects moving to Florida. Now, of course, the vast majority of them will not cause any issue. They won't have a population that's large enough to get established. Perhaps climate will not be suitable. But from time to time, you've got something that looks like this. This is an ambrosia beetle. It arrived first in Charleston back in, I think, 2002. It's been detected in 2009 all the way down in Miami. And the problem is that this thing carries a fungus that kills every single tree from the Lauraceae family. All right? So every single tree within that entire area that I was showing you there before is essentially dead. All right? There's no resistance to it. Okay, so how do you address this kind of emergency when you have this type of pathogen that invades your area of interest, and how do you respond to that? I mean, obviously the approach would be through breeding, but if our breeding is going to take a quarter of a century, we probably will not stand much of a chance. And of course, we've gone through this before here in the U.S. with the American chestnut, for example, which are populations were decimated in the eastern area. So we got to do something to do this a little bit better. And as I was mentioning before, people that really do the field work in breeding, like Tom, they have come up with very clever ways of accelerating, for example, the breeding stage. So we do something that we call top grafting, where we take a selection, we graft it into an adult tree, and then we can make that adult tree produce flowers very early on. Okay, so this is one approach by which you can accelerate the breeding process. You can also propagate your best material using things like somatic embryogenesis and rooted cuttings. Okay, so the big bottleneck has really remained the testing phase. How can we tell if a certain material is really good? And this is where we step in, because what we think and what we would like to do is really use the molecular information to make that selection very early on, so that we can go from a cycle of breeding that used to take between 15 and 25 years to take only 5 years or less. Okay, this is what we're shooting for. All right, so this is where genome-wide prediction or genome-wide selection comes in. So genome-wide prediction or genome-wide selection essentially is based on a relatively simple principle that really Winston outlined back in 2001 in that famous paper in genetics. And the idea is this: you've got your population, and this is what we're going to call a training or a discovery population, which could be actually your actual breeding population. Okay, you phenotype and you genotype that population, and then based on that information, you develop your prediction models. And those prediction models, they are somewhat different from our traditional QTL analysis or association studies, in that we really try to fit all the molecular information in predicting the trait. Okay, so there are a number of statistical approaches that one can use to do this. I'm not going to go here in detail about them. I'm just going to show you a few results that show you the comparison about the performance of these different methods. But the idea is that after you develop that prediction model, you break down your population and you use that model to predict a subset of that population. In other words, you do what we call a validation step, a validation process. Okay, and if that model is good, then what you have is a multi-locus model that predicts your phenotype. So if you develop this at one given cycle of breeding, the idea is that, of course, you can use that across multiple generations to make selection. The principle behind genome-wide prediction is actually relatively simple. Let's assume we have the blue bars here represent 5 different chromosomes, and let's assume our green boxes here represent regions that regulate the target trait. Okay, they're QTLs. The idea is that if you have enough markers to really cover this genome to a great density, you will always have a QTL that is in linkage disequilibrium with one of your markers. Okay? In other words, you're really trying to capture all that marker, all that phenotypic information based on the markers that you have. It's relatively, really, in a certain sense, a relatively simplistic approach. And of course, in most of the cases, the markers that you will be working on are not the causative polymorphisms, right? They are not the actual markers that cause the difference in the trait. They're simply markers that are in linkage disequilibrium with the causative polymorphism, all right? So we're not claiming here that the SNPs that we use in our prediction models are actually the SNPs that regulate our traits. Now, of course, then as I mentioned, the idea here is that if you have your prediction model, you can now begin using it when you develop materials out of your training or discovery population. You essentially apply that to the first generation to make a selection of what are the best individuals based on their genetic makeup. Okay? And, of course, you can apply this to 2, 3 generations or more. We're going to talk a little bit about that in a few minutes for how long you can really apply this kind of information. Okay? So, one of the issues here is, of course, when I develop my prediction models, a lot of times people approach me and say, well, great, so now we can use this prediction model for 1,000 generations. Well, unfortunately, it's clearly not the case. The number of generations that you can apply this prediction model really depends on the number of markers that you used and the degree of association of those markers with the genetic polymorphisms, okay? So if you have, let's say we're looking here at one chromosome, and we have these different haplotype blocks here in green and red, and we have our QTLs here, and what those arrows represent are our molecular markers. Okay? You can see that in this case, this marker over here essentially is in linkage disequilibrium with this QTL, this marker is in linkage disequilibrium with this QTL, and so on. Okay? I'm fully explaining my model based on these 1, 2, 3 markers is fully explaining my QTLs. Now, as we move to the next generation, what you're going to start having, of course, are recombination events that will break down that association between marker and QTL. Your models will become less and less accurate. And over time, of course, the expectation is that your model will no longer be valid to be able to do a prediction. So what you see a lot of people in cattle breeding doing or in poultry breeding doing is actually recalibrating their model either every generation or every 2 generations or so. Okay? So this is not a magic bullet that you develop your prediction model and you're going to be able to use it for 100 generations or so. Okay? Now, one thing that we do have to remember is that if you do have a larger number of markers, what's going to happen is that you no longer have just a single marker associated with your QTL, you have multiple markers. So even if you have a recombination event here, you still have an association over the next generations. In other words, the more markers you have, the more likely that your models will remain relevant for multiple generations. Okay? So of course, numbers of markers here are a pretty critical element in this whole thing. Okay? So, all right, we develop our prediction model. Is it good? Is it not good? How accurate is it? Fortunately, we have a number of somewhat rules that can dictate the likelihood that our prediction models would be good or bad. Okay? The accuracy of a genome-wide prediction model really depends largely on the extent of linkage disequilibrium in a population, on the training or discovery population size, heritability, and the number of QTLs controlling the trait. This all makes perfect sense if you think about it. Okay? Think about the importance of linkage disequilibrium in defining the accuracy of your prediction model. If you have a population with a very small effective population size, your blocks of linkage disequilibrium would be relatively large. Therefore, your likelihood of having markers that are associated with a QTL are quite high, right? That would be the expectation. When you have a population which has a very high effective population size, then these blocks of linkage disequilibrium are very small, and therefore your likelihood of having markers that are associated to traits is low. Okay, and therefore the expectation would be that your accuracy here would be a lot lower. Now, the very positive thing about this whole story here is that as breeders, we can control the effective population size of our breeding populations. Okay, in other words, we can really manipulate them to make them suitable for genome-wide selection if they're not suitable yet. Okay, so we can really work around with that. The other thing, which is an important factor, is of course the training and the discovery population size, and it sort of again makes total sense. The more individuals you have, the more precise will be your estimate of your allelic effects, right? So with more individuals, more accurate the story is gonna be. Just from the perspective of what we have been doing at the University of Florida, we are targeting populations around 2,500 to 3,000 individuals, and the reason for this is because our data on the initial generations of genomic selection we've done, they agree very well with these empirical studies that have been done. Actually, these simulation studies have been done, which show that the accuracy seems to plateau to some extent when you have roughly 2,500 to 3,000 individuals. So that's essentially what we have been targeting in our breeding programs. Effective population sizes of 40 to 60 with training population sizes of a couple of thousand to 2,500 individuals. Okay? But again, this will depend heavily on a number of factors, number of markers that you have, and your expected number of QTLs, which of course is not a number that you can know, but is a number that you can guess to some extent, or at least have some kind of estimate. Now, of course, not unexpectedly, heritability, right? The more heritable the trait you have, and again, this is data from an analysis that we did of 17 different traits, where we looked at the narrow sense heritability estimated for that trait, and we looked at the predictive ability, and we saw that there's a very nice correlation. Of course, the higher the heritability, the higher your predictive ability. Okay, and I just want to, for those that are looking at genome-wide selection papers, you will see that sometimes people refer to accuracies. Some people sometimes refer to predictive ability. Some people sometimes refer to correlations. Just sort of as a disclaimer, when we talk about accuracy, we are really dividing the correlation between the expected breeding, the breeding value, and the genomic breeding value by the heritability. Okay, so that's why usually values for accuracy are much higher. Okay. Now, and then finally, the number of QTLs controlling the trait. As you can expect, if you have a very, very large number of genes regulating your trait, your ability to capture their effect in a predictive model is lower than if you had very few QTLs controlling the trait. So, from what I just mentioned to you here, the very positive thing about it is that there are several steps in the design of an experiment, in the design of a study using genome-wide selection, where the breeder has control. Okay, you can control effective population size. You can control, to some extent, the heritability of your traits, depending on the design of your study, and you can control the size of the training population. Okay, so you really have the power to control, to some extent, the accuracies that you will obtain in these kinds of studies. Now, this, of course, the whole buzz about genome-wide selection started roughly, about 10 years ago. I mean, of course, Winston usually first introduced the idea, but by the time he introduced, really, there was no way you could genotype thousands of markers at a very low cost. So, of course, the cattle breeders took on this the moment they actually had tools and approaches by which they could genotype thousands of markers. And that was more around 2004, 2005. That's when really this began being widely adopted, especially by the cattle breeding community. And there are some countries today where over 50% of the selections that are made, they're made purely based on the genetic makeup of individual, not based on any other type of information. I think actually the USDA catalog that you can acquire to essentially tell you the value of a given bull is today purely defined based on on the genetic makeup of the bull. Okay, so this really is a method of breeding that took off in cattle breeding. Plants were a little bit slower to adapt, and I think there's a couple of reasons for this. I always say, at least in our field, most of the big advancements really have been borrowed from the animal breeders. I think the animal breeders, they work with such a restricted system that is so complicated that they really sort of have a certain level of pressure that we don't have in terms of creating novelty in terms of their breeding. Okay, so a lot of the things really have been inherited by the cattle from what the cattle breeders used to do. So we became interested in genome-wide prediction around 2007, 2008. And when we looked at what we had in our breeding programs, we suddenly realized that we actually had the perfect data set, the perfect population to actually carry out genome-wide prediction. We had one breeding population that we call C clones. I'll show it to you in a few minutes. That was composed of about 900 individuals. So again, we're not getting to the 2,000 individuals, 2,500 that we wished, but this is a relatively large training population. It had a pretty manageable effective population size. It had already been phenotyped for a number of different traits, and it had also been quite extensively genotyped with molecular markers. Okay, so we had roughly 4,800 markers that were available. The other really interesting thing about this population was that it had been clonally replicated, so we had replicates in 4 different sites across the southeastern U.S. Okay, this in forestry is rather unusual that you would actually clonally replicate a breeding population, but in this case we had this available. So we put this information. We had a student that was just arriving from Brazil with a lot of experience, and we took on and we began developing prediction models to essentially see what they would tell us. Okay, this just gives you an idea of that training population. It is essentially composed of 32 parents. They're here in the row and in the column. And every time you've got a cell that is filled, it means that there was a cross between, for example, this individual and this individual over here. We got the number 18 here. It means that the progeny size from that cross is 18. Okay, so roughly we had 900 individuals here derived by crossing these 32 individuals, and this design is a connected circular mating design. Okay, again, they are sort of maybe a little bit unique to forestry. What you've got here at the bottom is actually the IDs of these families. But what Marcio then did, and Marcio has actually a lot more hair now than he had at that time, so you probably won't recognize him around. Marcio, he took the information that we had for the trait diameter at breast height as well as height, measured in these 4 different sites, okay, that I showed you in the slide before, BF Grant, Cuthbert, Nass, and Palatka. And he then developed a prediction model using a standard approach, ridge regression BLUP. Okay, I'm sure most of you are familiar with BLUP. So that's the ridge regression approach to it. And he calculated the accuracy of genome-wide prediction in that one given generation. Okay, the accuracies that we obtained, they were pretty much in line with what we were expecting based on those simulation studies that we were looking at before. But here's the really interesting thing: is that you can actually estimate what would be, based on your heritability, what would be the accuracy of traditional BLUP breeding in the same set of individuals. Okay, and what we noticed is this. Well, first of all, in general, it is true that genome-wide selection is giving us a slightly lower accuracy than BLUP-based breeding. However, if you assume that now you're increasing your rate of your breeding cycle, you're doubling the speed of your breeding cycle, the actual efficiency of breeding using genome-wide selection was significantly higher than if we were using traditional breeding. Okay, so again, this is a population that was not designed to be a population to be used for genome-wide selection. It was a population where we had data on. We did not have the number of markers we wish. We wish we had about double the number of markers. But we already had an indication that very clearly the accuracy that we could obtain were actually quite good. Okay. The other question that we were interested about, this was the work of Patricio, he's now a faculty at UF. One of the things we were interested about was, okay, what happens if you now use traits with different types of genetic architecture? Okay, if you work with a trait that's oligogenic or if you work with a trait that's polygenic. And we compared several of the different methods from ridge regression BLUP, Bayesian lasso, Bayes A, Bayes C-Pi, to see how accurate they would be in predicting these traits. Okay? The overall conclusion from what you can see in this table is that there's not a heck of a lot of difference between the methods. to develop your prediction model. Okay, the reality is that these days we use primarily Bayes CPI. One of the reasons for that is because it adapts itself quite well to traits that are oligogenic in nature. Okay, and rust is a trait typically controlled by a few genes of large effect and a lot of genes of small effect. So it's really a trait that's oligogenic. And actually, Bayes CPI does quite a bit better than the ridge regression blood, for example. Okay, and here you just get an idea, essentially, of the number of markers that we need in our model in order to be able to reach the maximum predictive ability. So this is actually times 10. So when you see 100 here is actually 1,000, 2,000, 3,000, 4,000. And you can see that for you to reach maximum accuracy for diameter, you need more than 1,000 marks, okay, in your prediction model.
What's the genetic map length?
The genetic map length is about 1,300 centimorgans. Yeah. For rust, which is the trait I was mentioning to you, is an oligogenic trait, you can see that the actual maximum predictive accuracy is reached with a far smaller number of markers, okay? And actually, after that, the model actually decays. It probably start having problems with overfitting the model, okay? So clearly, different architectures will respond in different ways, so you need to be aware of what you expect in terms of the traits you work with. But then, another question we were interested about to respond with this data was how accurate are these prediction models across environments? Okay, I think one of the concerns that we've always had was that breeders sometimes would approach us, say, "Well, great, so you developed this prediction model for this population. Now I'm going to use it across the southeast. I'm going to use it across the country." And genome-wide selection is just like traditional breeding. You develop a selection process in a given location. If you move to a different site, it's different. You're going to have environment by genetic by environment interaction that will mess up your selections, right? So what we did was this. We used the data that was collected in each one of the individual sites to develop the prediction model, and then we validated that with data from the other sites, okay? So what you can see here quite clearly that if you are estimating your model based on two sites that are very close together, you can see that the model actually performs quite well. For example, Palatka here performs quite well in Nassau, in data from Nassau. Nassau performs quite well in data from Palatka. Okay, so the model is good. You're applying it to the same breeding zone. Okay, now you use that same model developed for Palatka. You try to use it to make a prediction based on data coming from the F Grand, it's northern Georgia. You see that the accuracies essentially fall apart. Okay, which is pretty much what you would expect. But it's good to be aware that this is not a magic bullet. You still, genotype by environment interactions are still really a very, very significant effect. So just to summarize this first part, we were quite happy to see that the accuracies are overall in agreement with what is expected based on the properties of the training population, the trait genetic architecture, and the genotyping density. In other words, we are reaching within this generation the accuracies that we were expecting to achieve. The other point here, again, as I said, genome-wide prediction is just like traditional breeding. Don't use a selection and breeding zone that's different from where the model was developed. But then, of course, many questions remain, right? We really didn't run this prediction over multiple generations yet, because our next generation is probably going to happen only 5 or 6 years from now. Okay, so how will these models behave over multiple generations is a big question. One thing to remember is that the cattle and the poultry breeders, they're constantly recalibrating their models. Okay, so you see very often that they will, at every generation or every 2 generations, essentially rephenotype the population and then redesign the model, rebuild the prediction model. And then, the world of prediction models is really saturated with noise. Okay, you have pretty much one new way to develop a prediction model coming out every year. It's more or less where we were with QTL analysis 20 years from now. If you recall, you would have essentially a new paper on a new method to map a QTL coming out every couple of weeks, right? We are essentially at the same place right now with genome-wide selection. That's part of the reason why, for example, the Journal of Genetics They use this data set as a standard data set where they say if you approach the journal with a new method, you have to prove that that method will do better in terms of your prediction model than previous methods that have been developed before. Okay, so clearly you can create your own Bayesian alphabet for your prediction model. Okay, if you tweak things a little bit here and there, there's certainly a lot of room to make improvements, but really significant improvements. There have been very few really out there. Okay, all right, so. One of the problems with genome-wide selection or genome-wide prediction is that they were developed primarily by animal breeders, right? And animal breeders, especially cattle breeders, what they really care about are additive effects, right? Because they want to take that bull, make a prediction of how good that bull is, and then they want to inseminate 1,000 cows with it. They really don't care what is the specific combining ability of that bull with a given cow. Okay, our situation is a little bit different. We know that dominance effects are important. We want to be able to estimate the specific combining ability, the ability of crossing two specific individuals and getting a good family. So we would want to also be able to predict dominance, not just additive effects. Okay, so. The reason why we would like to do this, of course, is because incorporating dominance and non-additive effects, we could potentially improve the accuracies of those prediction models. And what we also think is that if we use that marker data to partition the variance that accounts for the phenotype, we may be able to understand a lot more about additive and non-additive effects that contribute to traits. Okay? And that is a matter of significant debate. Of course, in the quantitative genetics literature, how important are additive effects, how important are non-additive effects. We see from our data and from our populations, we think that non-additive effects are quite important, and we should take them into consideration. So, why using genetic markers in order to develop relationship matrices? How should that be better? So much of the breeding out there is done based on methods like BLUP, where you need a relationship matrix, right? And if you have only pedigree information, if you don't have marker information, then your expectation relative to resemblance of relatives would be that for the additive resemblance, for an additive relationship, it would be 0.5, 0.5, and 0.5 between these individuals. For dominance, you would expect it to be 0.25, 0.25, 0.25. But in reality, what's going on in the genome of these individuals is such that they may not share 0.5, a relationship of 0.5. So if you have your marker information, you actually might be able to derive precisely what that relationship truly is. Okay? And you can simply think about a situation where one individual is fully heterozygous, and that individual transmits one half of its genes, of course, to one progeny and the exactly other half to the other progeny. Okay? You would expect that their genetic relationship is quite limited. Well, if you have markers, you can actually account for that. Okay? So you may find out if you use your markers that that relationship is actually 0.52, 0.43, and not exactly 0.5 or not exactly 0.25. Okay, if you're talking about dominance. Right? So if you have markers, again, you can estimate precisely that relationship. And this is what the data actually looks like for C clones. Here you have the relationship derived by the pedigree, and I'm sure you cannot see the numbers here, but I can tell you that this is 0.5. This is the relationship derived from the markers. Again, this line here is 0.5, but you can see there's quite a bit of variance around the number 0.5 here. Okay, so though we were assuming that the genetic relationship was 0.5, in reality it really deviates quite significantly from that number. Same thing for dominance. Okay, if you apply a dominance matrix here, you can see that the number will vary quite significantly around the number of 0.25. Okay, so the markers can be quite useful in terms of deriving that genetic relationship matrix. So what we thought was this: all right, let's build models where we try to predict height, where we include additive effects based on pedigree, additive and dominance, additive dominant epistasis, based on pedigree, and based on markers. And let's see how well these different models can partition the genetic variance, okay, into its different components. And then, of course, also evaluate the prediction accuracy for the trait tree total height. Okay, so this is where things got really quite interesting. What you see here are the models where include additive and dominance effects, additive by additive, dominance by dominance, additive by dominance using epistasis. You can see that the heritability is consistent across the models, and they're all based on pedigree. Okay, none of this is based on markers. Okay, dominance also remains constant across the population, and the accuracies remain pretty much the same. When you now include the genetic marker data, you can actually see that not only the accuracy goes down, but especially the heritability drops significantly, and an effect of epistasis seems to appear and seems to be pretty significant. Okay. Now, this is not the ideal population to test this. Okay. The right place to test this in a conclusive manner is really using simulations. Okay, and this is what we're doing actually right now. We're building populations, we're simulating populations that would look like the C-clones population, including dominance effects, including epistasis, and really trying to see if that confounding effect of epistasis and dominance with the additive effect is really exists or not. Okay, but this is the main point I want to make here, is that after we include dominance effects and after we include the marker information, we saw a very dramatic decrease in the heritability. Okay, in other words, that additive variance, the data we have suggests that the additive variance that we are estimating is actually heavily inflated, and that actually the true additive variance is a lot smaller, meaning that our estimates of heritability are inflated, and meaning that our estimates of gains are inflated. Okay, so what we see markers playing a role here is really in allowing us to break down that genetic variance more precisely, better into its different components. I'm sure that everybody that had classical quantitative genetics, you learn that the additive variance is orthogonal from your variance coming from dominance and non-additive effects, right? I think what we see here is that this is not true. These two things are not independent, and you really need ways to separate them properly, because the additive variance is really confounding, is really being confounded by other sources of genetic variance. Okay, so just to summarize the second part here, additive and non-additive effects may be confounded when they're estimated based on the pedigree. We think that the marker-derived relationship matrices, they produce a better partition of the genetic variability into additive and non-additive effects. The implications, again, as I mentioned, is that we could be overestimating additive effects, which would then mean that we're overestimating our heritabilities, and as a consequence, overestimating our expected gains. Okay, simulations are needed. Okay, and that's currently underway. The only problem is that if you may know that the pine genome is about 7 times the size of the human genome. The size of one pine chromosome is the size of a maize genome, more or less. So to simulate that, there's no simulator out there that can actually handle this kind of data. So we finally got it to work a couple of weeks ago. So these simulations are finally now on the way, and then we're really going to be able to throw in additive effects, different additive variants, different dominance variants, to really see how well our models are truly being able to pick this up or not. Okay, so obviously this is a critical part in this analysis. But why? I mean, other than just developing models that are more accurate, that capture more than just the additive variance, what else? What else would we be wanting to do this? One of the reasons that we want to develop these models is because we want to do what we call mate pair allocation. Mate pair allocation is the idea that you would be able to, from your prediction model, precisely identify which individuals you should cross in your population. You should be able to identify not only the mean family values, but also the family variance of every single family you could potentially generate from that cross. Now, if you assume that you only have additive effects, you just basically need to average your values, your breeding values of your parents, right? But when you have non-additive effects, then your models need to account for those other effects. So the question that we asked ourselves was, for example, here in C clones, we've got roughly 900 individuals. And what should we cross? Right? Should we cross an individual in this family with this one? I mean, we have way too many possibilities. And in some cases, we may want to identify a cross that will have a superior mean family value. Okay, like in this case, mu2 is bigger than mu1 here, so we would favor this cross. But in some cases, perhaps the breeder don't just want a better or higher mean family value. It actually wants bigger family variance, because if you have bigger family variance, you have a higher probability of picking up things that will be in the extreme of the phenotypic distribution. Okay, so we're not just trying to figure out which family is better, but we really want to try to figure out what is the variance within all these different families. Okay, so we began with this idea of surgical breeding, whatever title you want to use to make it sound really fancy, and we said, all right. So now if we take every single individual and we simulate a cross based on all the markers that we have, we can predict that mean family value. The variance is a little bit more complicated, and we're really still working on it. But then essentially we can contrast this one with perhaps another individual and see if that mean family value is better or not. Okay, and this is something that, again, as I said before, is something that makes sense doing if your model is capturing something else other than just additive effects. So this is what the data looks like. You have 900 columns and 900 rows. Okay, what you have in each one of these little pixels here is the predicted cross based on the mid-parent average. In other words, you're accounting only for additive effects. And here is your predicted value based on additive plus dominance effects. Okay, and you can see that these two matrices are not, these two heat maps are not the same. Okay, the correlation is 0.82, meaning the effects of dominance, they really changing ranks in the way we score these families. Okay, so in other words, they really can be very significant in the sense of how they're going to guide what we select for breeding. So this is how I see us using this type of information, and we're using this information now to guide a few crosses that we are doing in the context of a USDA funded grant. We said the following: all right, one of the things that we may want is a family that produces a very good phenotype for a certain trait like volume. Okay, but perhaps another thing that we may want is a family that will perform very well across the entire southeastern U.S. In other words, this family has very high adaptability across the region. That would be very valuable for breeders, right? You don't have to plant very specific families in each location, but rather you use families that are suitable for that entire region. So what we did was to develop the prediction models for each one of the individual sites, including additive and non-additive effects, and then we made a prediction of the mean family values of all those 900,000 possible crosses. Okay, no, 500,000 possible crosses. So what you have in these histograms here basically is the different values and the different families that could have been generated in Palatka, Nassau, Cuthbert, and BF Grant. And there's one family down here that is predicted to perform very well in Palatka, perform very well in Nassau, perform very well in Cuthbert and BF Grant. In other words, it is a family that we predict to have very high performance for height across that entire range that goes from Florida to northern Georgia. Okay, so this is one of the things that we have made crosses for, and of course now we're going to have to wait about 5 years to get this heat. right? To be able to tell whether they're truly better adapted to the environment or not. But we also pursuing other types of crosses that are more dedicated to specific end products, and one of our goals is to produce families that are more suitable for biofuel production. So in this case, we're looking for families that will have a predicted low incidence of rust, low amount of lignin, high cellulose, and high diameter. Okay, so what you see here again is the distribution of all the different possible families that could have been created for rust, lignin, diameter, and cellulose, and we identify one specific family within these 450,000 possibilities that has low rust, low lignin, high diameter, and high cellulose. Okay, so these crosses are now being made, and the goal is the moment we have these families, we can essentially be able to test whether if they indeed have low rust, low lignin, high cellulose, and high diameter. So it's a very, very, very targeted, very specialized, let's say, type of cross. All right. So to make a long story short, this is how I see that this is probably going to have an impact, at least for us in forestry. We are in a situation where we need to address challenges much faster than we can do through traditional breeding. Okay, what if those challenges come from climate change? What if they come from changing products? You know, terpenes are something that nobody has bred for many decades, and now suddenly everybody is interested about it again. And changing threats. Okay, we need to be able to address these challenges by breeding for them. The idea is that you can have a training population that at any time one of these threats or these new needs come up, you train that population, you identify, essentially, you phenotype that population for that certain trait that is related to that challenge, and then you design the crosses that will produce the families that will address these different needs.
What if you don't have the trait in level 3?
Oh, then you're in trouble. All right, you can't make any miracles like, you know, it's like the citrus breeders. They really haven't paid too much attention to genetic diversity, and now citrus green is essentially eradicating the culture. Yeah, I mean, definitely you won't be able to address it, right? But our intention is, you know, from going from this to going to this a few years ago, is to really come down to something that would look like this, where we would be able to really be carrying out the generation of breeding every 5 to 6 years. Okay, this would truly represent a revolution for forestry because, again, it would mean a very dramatic gain in efficiency of our breeding programs. But again, it's something that really is now at the stage of being evaluated. Okay. I think I'm right on time. So now again, these are the acknowledgments again. There are a couple of other people that I added here. These folks all came to the Science Without Borders. Brazil is a constant supply of marvelous quantitative geneticists to my lab. So a lot of them have worked out very well. We have a lot of them that come to visit. And again, these other collaborators. And most of the work that I talked to you about right here has been supported by this grant from the USDA plant breeding program. Part of this grant is training of breeders in genome-wide selection. Okay, so one of the goals is to develop a module where essentially people will be able to become more acquainted about the methods of analysis. But we also organize one workshop every year through this grant. We just had the workshop about a month ago. We had over 600 people registered to attend it online, okay, from 58 countries, which sort of shows you the interest in genome-wide selection that we have right now. So we had Rex Bernardo giving a talk, we had Guilherme Rosa giving a talk, as well as Marcio. So again, it's an exciting time, I think, for breeders in terms of using this kind of technology. I think there's still a lot of questions that need to be answered, but I tend to, just like the breeders before me in forestry, we have had a tendency to do a lot of what the animal breeders have done before us, and the fact that the animal breeders have really integrated genome-wide selection so effectively into their breeding programs really gives me some hope that this will truly have an impact, at least in the cultures that we work with. Okay, and I think that's what I had. Yes. All right.
So I know some students probably have to get to class, so don't feel shy about going out, but hopefully Matias can entertain some questions. Got 15 minutes or so.
All right. This was either very confusing or you guys already knew everything.
I have a
Yes,
simple
go
question.
ahead.
I got a question. If you were to predict a trait that has more genes that control that trait, so mutate them, on your graph where you have your power of prediction by heritability, would it be to this side of the graph, right? With less power to predict.
If you had a lower heritability?
Yes.
Yes, absolutely. Yeah.
So how would you go around that? Because we work with crops. I understand
Yeah.
that your yield is based on height,
Yes.
but still height.
We typically work with traits. Forestry, you complain about crops. Heritabilities in forestry are awful, generally. Okay, they're typically worse than in crops, largely because our ability to really control the site is somewhat limited, and because these trees will grow over multiple years, they're going to be challenged by the environment perhaps much more extensively than if you grow a crop for a few weeks. The heritability, it really depends on your population. I mean, if you have no diversity for a given trait, your heritability will be 0. So you have to, it's the same challenge as it is in breeding, right? If you have no diversity, if you have a very narrow genetic variance, your heritability will be low, you make low gains, right? So I think in that sense is no different than traditional breeding. And just like in traditional breeding, if you want to make a suitable selection, you really need to pay a lot of attention to the conditions of the trial, because you want to minimize, of course, environmental effects, and that's another thing that can contribute to improve your heritability. So there's some that you can work with, right? Carol, you had a question?
Okay, yeah. With regard to this slide, so you showed the picture of the factory that burns the pine to make the bioenergy. Is your breeding scheme going to be the same no matter what method the bioenergy will be produced? Like,
Well,
are you thinking about ethanol production or
right.
something else? And is that
it's
where
going to so it's going to depend on what they want to what the final product of interest is, right? If they want to produce, if at the end of the day what they want is terpenes, then we probably want low rust, high growth, and high terpenes. And then we will identify the crosses that will produce that are predicted to produce that outcome at the best possible way. If, for example, that factory that mill essentially is just burning wood, in that sense, maybe if you have more lignin, it's actually going to be better than if you have less lignin.
Yeah. That's why you were putting in low lignin.
Yeah. Well, here's because we were thinking about the scenario producing biofuels. Okay, so we want to ferment that and really be able to extract the cellulose and make the ethanol from it. But yeah, I mean we can. Yes.
If you're a public breeder, many of us are, in an annual crop, and you're into this genome-wide selection model, how much of your time is this? How much is making these training populations and determining the model going to be required? Is it going to take up half of your time, 25% of your time? I'm trying to get to something. How much? What am I going to have to give up?
What kind of personal investment? Yeah. So I think there's two elements to it. There are the things that you already do anyway, which are phenotyping and so on, and then there's the element of genotyping, and there's the element of running the models, right, and selecting what you want to do best. I'm very much against the idea of genotyping in-house. I'm very much into the idea of outsourcing that because it's a technology that's changing so rapidly that when you just choose to make an investment in something, next year is going to be half the price, and you made that investment. It was a lot. You lost a lot of money in it. So I don't think you should spend any personal investment in that step of the genotype. I think that should be outsourced. The analysis per se, in a sense, is if you already have someone that's running BLUP that is already doing selections, it does add very little to their. This is not something there are a lot of packages out there, and for example, we're releasing one including dominance soon that really pretty much automate this whole process. And quite amazingly, they're not computationally as intensive as maybe someone would expect. So there's a lot of ways by which you can do this in a very efficient way. I think, of course, the biggest cost there is the genotyping, right? You're adding that cost. And from our, for tree breeding, it makes a lot of sense because the gain in time is tremendous. But what I have seen when you talk to the big seed companies, it appears that a lot of their interest is not necessarily in accelerating gain, because they're already breeding 3 or 4 cycles a year, right? Their biggest interest is in the fact that if you use enough markers, your accuracy of selection is higher than your phenotypic selection, right? So you actually are more accurate in your breeding than if you were simply phenotyping the plants, right? So a lot of entities for which doing early selection is not as valuable as it is in forestry are making the argument that it is valuable for them because of the gain in accuracy.
And of course, big companies can throw a lot of people
Certainly,
at it.
yes. Mm-hmm.
The big expense may be in the genotyping. Well, I would think the biggest time commitment and expense would be in the phenotyping.
Yes.
Because you got to go back out now. I've got to go out every 3 years or whatever and phenotype 3,500 individuals.
Yes. Yeah.
Populations, which is a pretty good.
Yes,
commitment.
it is a big commitment. Yeah. So
if you're trying to be prepared, as your last slide, to have all these phenotypes for bugs or diseases or whatever else.
Yeah. So from our perspective, because genotyping is still relatively expensive, our approach is to phenotype for as many things as we can. Right, because the phenotyping is still not as expensive as the genotyping, but there will be a time in which, of course, the phenotype will be the restricting factor. However, I think the same or similar types of gains that we have made in genotyping, making it more affordable and faster, higher throughput, will probably or will have to be achieved on the phenotyping side. I think there's a lot of recent publications from the NRC and so on suggesting more and more that phenotyping is now the bottleneck. This is where a lot of funding should be focused on in terms of making it more efficient. So I think I hope that side of the phenotyping side will become more cheaper and easier to do. Yeah.
How difficult is the retraining and rearranging of the population when we deal with the pathogens such as thrust, which changes dramatically fast because of mutations? How difficult is that?
I don't know. I don't think anybody has. It would be interesting to see if anybody is applying. I mean, from my expectation, probably the most logic thing would be to challenge with a new pathogen every time you have a new strain that becomes a dominant strain, right? Whatever that's feasible or not, it's something that probably depends on the pathogen.
Yeah, especially in our case, I work on wheat and you have to get these genes from not the wheat itself, but from relatives. Associated with a lot of poor quality and lack of adaptation, so you have to include those lines and retrain the population. It's just another.
But I think, but think about it. You're now doing it based on molecular data that is tracking specific loci that are linked to your gene of resistance.
You're also getting a lot of genetic drag.
Right, but you can control that genetic drag much more efficiently if you have markers distributed across the genome, because then you use selections, you can minimize the drag of that genetic material you don't want from the loci of the alleles of resistance that you do want.
So pluses and minuses.
Yeah. I think there's definitely.
I don't think you mentioned how many markers you have in this population, but to follow up on Wayne's question, maybe you did. But
I had 3 markers per cent. Well, so 3 markers per cent Morgan. The map is about 1,300. We had roughly about 4,800.
4,800 markers. Okay. So if you throw all that and your 900 individuals, 4 locations, you throw that into your analysis program. I'm assuming you got your code well written now. So to get 17 traits on all locations and give you your prediction, what type of time are we talking about now?
Less than a day.
Less than a day.
Yeah, on a laptop. On a
Really?
Yeah, using ridge regression. Okay, there'll be some more sophisticated ways of doing it, but using ridge regression is relatively fast.
Any students with questions?
I have another question. Why this has shown a lot of promise in animal breeding compared to plant breeding? We have seen a lot of data from
Yeah. So I have no doubt that the reason is because if they can artificially inseminate and genotype an embryo and don't have to wait 3 or 4 years for that cow to become productive, it is a significant gain, especially because that animal, the cost of raising that animal, feeding that animal, treating that animal is in the thousands of dollars. And therefore, the advantage of using this kind of technology to make that kind of to make a selection is huge. All right. So in other words, they have two issues that come into play here. One is time. It takes cattle breeding does take time, and cost. Right. The cost of raising every animal is quite high. In forestry, we have the issue of time. Unfortunately, our trees are too cheap. Right. I wish they were $1,000 each. We would be doing a lot better if they did. But unfortunately, they cost a few dollars only. So really, from our perspective, the biggest advantage in terms of time. So I think every culture will really have to make a very careful assessment of what kind of advantage can it bring to me, to my breeding program.
And the point I try to make is, because it has a fancy name doesn't mean that to be the best option for every program.
Absolutely, absolutely, and I certainly urge every. I think the one thing that is important for the breeders, especially for the students that perhaps are sitting here, is you need to be aware of it. You need to be able to communicate about it, because I think more and more companies will approach you and will ask you, well, okay, you want to work here, but do you think we should do this? Right? Do you think it makes sense? So I think. I'm not claiming everybody should be trained in this because, it may not make sense, but to really be able to communicate and make a decision as to whether if this should be something to be pursued or not, I think it's really relevant in students' training. It would be useful.
Well, let's thank Matias for an excellent start. And just a reminder, 3 o'clock in room 303 of the horticulture and what's your building? Horticulture and forest
Science.
Forest science building.
And if you're sitting in a chair that looks like it doesn't belong in